topic that we'll be dealing is about the complement system. So complement system gets its name by the term complement. That means this system complements the function of antibodies in bringing about the lysis of the bacterial cell. So this complement system are nothing but consists of wide variety of complement proteins that are present in the serum. So they are also called as serum components that usually circulates in the serum in an active form, and upon antigenic challenge and by the formation of antigen antibody complex, these complement proteins get activated and complements the antibodies to bring about the lysis of the bacterial cell. So they are usually activated by antigen antibody interactions. When there is a specificity between antigen and antibody, the complements get activated. Before that, they are the one that circulate in inactive forms, and these proteins are usually heat labile component. So initially it was thought that there is certain component in the serum that is heat labile in nature, that the bacteriolytic activity is lost when these components are usually inactivated. So studies were carried out on this component in the serum by different scientists. It was Buchner who proposed that the bactericidal activity is present in the normal serum having the components which are usually heat labile in nature, and it was Prefer who has proposed as a concept of bacteriolytic activity, again with the same components that are present in the normal serum. And finally it was Bodded who said that bacteriolytic and hemolytic activity is present in the normal serum, and the serum contains certain components that complements the activity of the antibody. So apart from heat stable antibodies, other components are also present in the serum that complements the antibody to bring about the hemolytic activity or the bacteriolytic activity. So he had given the term called as alexins. Later on, these alexins were renamed by Ehrlich as complements. So now we know that there is a complement system in the serum of an individual that is able to carry out bacteriolytic, bactericidal and hemolytic activity. Now these complement protein are the one that usually carries out the biological effector mechanism by interacting with the antigen antibody complex or interacting with the different immune complexes that are formed in the human body. And these complement protein usually work in an enzyme cascade. This means that there are different complement proteins that are involved in this reaction, and one of the protein, once it gets activated, it becomes an enzyme and it activates the other complement protein till it brings about the lysis of that particular organism. So there are different complement protein getting activated one after the other in a cascade manner, and it is usually enzymatic in nature. So the function of the complement protein is to eliminate the antigen interacting with the antigen antibody complex and getting activated in an enzyme cascade. And this enzyme cascade is highly regulated, because if there is no regulation in this cascade reaction, there is a possibility that they may damage the immune system along with the pathogenic microorganism. So the complement system, though they are in inactivated form, they get activated only upon antigenic challenge. So the regulation is first at that particular time when there is an antigenic challenge, only then they get activated, and later on when the enzyme cascade is carried out at that particular time also, there is also certain regulation to inhibit the immune response to take place against a particular antigen apart from interacting with the self antigen. So coming to the general properties of these complement proteins, they are the one that resides in the serum, also called as the serum proteins, and they are the one that belongs to non-specific immune response, means they are the one that belongs to innate immunity. They are not specific towards a particular antigen, but they are the one that interacts only with the antibody that is in combination with the antigen. So the amount of these complement proteins that is present in the serum is about 5% of the total serum, and they are mostly heat labile in nature. It easily gets activated at 56 degrees centigrade. Mostly some of the components might be heat stable in a complement system, but overall when you look at this complement protein, they are mostly heat labile in nature. So it was the heat labile component that was first discovered, and later on the name complement was given to this, and they are the one that get activated only with the antigen antibody complex. So whenever the antigen enters, the first immune cell that comes forward to interact with the antigen are the antibodies. So depending upon the specificity, those antibodies are produced. Now when they interact in a specific manner, they activate the complement protein to get activated and interact with the antigen antibody complex. And depending upon how these complements are used to interact with the antigen antibody complex, you can define the terms to this complement protein, either that are said to be fixed, complement is fixed, or they are said to be complement binding proteins, or they can also be called as consumption, or they are consumed during an antigen antibody interaction. So mostly these complement proteins are effective towards the immune complexes. They are produced against the immune complexes, and the type of antibody that activates this complement protein are usually the IgM and IgG, that is a dimer. So it should there is important thing that has to be borne in mind, that only the multivalent antibodies are able to activate the complement system, but not the single antibody. Okay, so you need to have two antibodies, either it should be in a dimer form or a multimeric form to activate the complement protein. And coming to the components, there are 20 different types of complement proteins that are given different names depending on the function they carry out. So first, they are usually classified as C1 to C9 based upon their discovery, but not depending on the function. So the C1 to C9 are the ones that are involved in the cascading reaction, and apart from that, there are other nomenclature that are given as you have properdin, factor B, factor D, factor H, etc. So complement proteins are either given the name by the trivial names or by the other names also, and usually these complement proteins are enzymatic in nature, and each enzyme, once it has been activated, it activates the other protein of the complement system. Now once the enzyme has formed, it cleaves the other complement protein into two fragments which are of unequal lengths. One is called as a smaller subunit and the other one is called as a larger subunit. Take for example, if C component is getting cleaved, so it cleaves into C3A and C3B. C3A is a smaller unit and C3B is the larger unit. Exception here is that C2. C2A and C2B which are formed, C2B is the larger subunit that is involved in the complement system, and C2A is one which acts as an endotoxin. So irrespective of whether it is a larger fragment or a smaller fragment that is formed, always remember that the larger fragment is usually involved in the complement system, whereas the smaller fragments are the one that brings about the anaphylotoxic reactions. So complement activation, how they are activated, usually these complement proteins are present in inactive form, and they are induced only when there is an antigenic challenge and there is an interaction between an antigen antibody and antigen antibody complex is formed, and the nature of it getting reacted is called as a cascade reaction, and these complement protein acts as an enzyme to cleave the other protein into larger fragment and the smaller fragment, and these proteins, once they become enzyme, they are usually represented as a bar on their surface. That is, C bar means that it has become an enzyme to cleave another important component of the complement protein. Now, how these complements usually gets activated? There are two major pathways by which the complement protein gets activated. It is called as a classical pathway or the alternative pathway. So it is initial, you can say the initial induction, how these gets activated depending on which type of antigen or antibody is involved. This complement protein usually enters into that particular pathway. So classical pathway or alternative pathway. Irrespective of which pathway it is, both of them usually join together at C3b, and after that all the pathways leading to the formation of membrane attack complex will be same in both the pathways. So the classical pathway is usually activated by certain types of organism and antibodies, whereas alternative pathway is has its own activators. Now first I'll deal about the classical pathway. It is also called as a specific pathway. The name classical pathway is given because it was the first pathway that was discovered, and here we have C1 to C9 protein completely involved in forming an enzyme cascade reaction to form the membrane attack complex. So the membrane attack complex is the one that is formed which is called as the immune cytolysis. The membrane attack complex attacks the membrane of the bacterial cell, forms pores on the bacterial cell and kills the bacterial cell either due to osmolysis or the plasmolysis. So the first complement protein that gets activated in classical pathway is the C1 protein, and the other activators of this classical pathway can be aggregated immunoglobulins, or they can be DNA or C-reactive proteins. Now let us see how the pathway usually proceeds to produce the membrane attack complex. So it is the first complement protein, which is a C1 protein. Now C1 protein has three important subunits, which is called as the C1q, C1r and C1s. So the first C1q portion goes and interacts with the Fc portion of the antibody molecule. And you require two such antibodies. That is, as I told you, the antibody should be a multivalent one. Either it should be IgM or IgG. So the most potent activators of the classical pathway are the IgG antibodies. So the C1q portion goes and interacts with the 2Fc portion of the antibody molecule. Once the C1q portion acts on the Fc portion, it cleaves the C1r to form an enzyme called as serine protease. Now C1r being an enzyme, it activates another component that is of the C1, which is called as the CS. So C1 is having three subunits: C1q, C1r and C1s. C1q goes and binds to the Fc portion of the antibody molecule that has interacted with the antigen. It activates the C1r to form a serine protease, which in turn activates C1s. Now the C1s has a substrate on C4 complement protein. Now when this C1s acts on C4, it cleaves C4 into two components, C4a and C4b. As already discussed, the larger fragments is involved in the classical pathway or in fixing the complement, and smaller subunit acts as an anaphylotoxin, carrying about anaphylotoxic reactions or the immune reaction or the other biological effector functions. So once the C4b goes and binds, the C1s also have the ability to cleave C2 component. So the C2 component gets cleaved into C2a and C2b. Now the C2a here, as I told you, there is an exception where C2a interacts in the classical pathway, whereas C2b is usually eliminated. So now on the bacterial surface, we have this complex of C1,4b2a, which is called as C3 convertase. Now this means that this enzyme is not able to cleave the C3 component into C3a and C3b. Again, the C3a, C3b get cleaved by C3 convertase, and C3a is involved in the anaphylotoxic reaction, whereas C3b goes and joins this complex, which is called as C5 convertase. So here we have C1, C4b, 2a and 3b. More amount of C3a and C3b will be formed to produce more biological responses by producing C3as, and 3b's are usually formed joining this complex, which is a C4b, 2a and 3b. Now this is called as a C5 convertase because it is not able to cleave the C5 component of the complement system. Now once the C5 gets cleaved, again it divides into C5a, which is a smaller component, gets eliminated as an anaphylotoxic reaction, and C5b goes and interacts with the complex to form a C6 convertase. Now we have the whole complex of C1, 4b, 2a, 3b and C5b complex. Now this is ready to take over and cleave the C6 molecule. Now from then we have 6, 7, 8 and 9 molecule coming together to form the pore on the bacterial surface. So C6, C5, B6, 7 and then 8. So ultimately it is the 6, 7, 8 that activate the C9 component, and nearly about 10 to 12 C9 component usually joins the complex and forms or drills holes on the microorganism of about 100 angstrom in diameter. Now because of the pore that is formed in the bacterial surface, there is leakage of the content from the bacteria and ultimately the organism gets killed. So the whole complex from the C1 to C9 that is formed on the bacterial surface is called as the membrane attack complex, and the organism dies either due to swelling and bursting or due to plasmolysis, that is leakage of the content. So this is about the classical pathway. Now next, coming to the alternative pathway, as I told you, both classical pathway and the alternative pathway merge together at a specific reaction which is called as the C3 convertase. From then on, the pathway is similar in both the cases. So alternative pathway usually is also called as a Properdin system, and the activators of this alternative pathway is not an antibody, but there are certain components that are present on the organism such as zymosin, bacterial endotoxin, IgA, IgD, cobra venom factor and the nephritine. These are the activators of the alternative pathway. Here there is a spontaneous hydrolysis of the C3 component that keeps on circulating inside inside our body. So once there is an antigenic stimulus with this activator, the C3 component spontaneously undergoes hydrolysis to produce C3A and C3B. More amount of C3A and C3B are keeps on producing till they get stabilized by a factor which is called as a factor B. Now the factor B goes and joins the C3B to form C3BB. Now this B component has to be cleaved by another important factor that is present in the alternative pathway, which is called as the factor D. Factor D cleaves the B component into BA and BB. So BA is again released as an aflatoxin, and BB goes and join forming C3B-BB complex. Again this C3BB complex acts as C3 convertase to produce more amount of C3Bs. Now the larger unit C3B goes and interacts to form C3B-BB-3B complex, which is called as a C5 convertase. Now this is ready to convert the C5 into C5A and C5B. But before that, we should know that this complex is the one which gets easily inactivated. That is, it will not proceed further until and unless it is stabilized by a propyridine system or propyridine protein. Now the propyridine protein stabilizes the complex which is called as a C5 convertase. Now the C5 convertase cleaves the C5 into C5A and C5B. Again the next steps are the equal. That is the same as in the classical pathway. That is 5 activates the 6 to form an enzyme. 6, 5, 6, 7 activates the 8, 5, 6, 7, 8 activates the 9, and about 10 to 12 C9 proteins usually join the bacterial surface or the activates or the organisms that usually brings about the lysis of the organism. So now there should be a regulation, as I told you, because it is working in an enzymatic cascade. So there is a possibility that more and more amount of aflatoxins can be produced inside the body that can lead to severe damage in the human body. So there should be a regulation that should control the complement system getting continuously activated. So this is called as the exhaustion system of the complement system, and if this is not regulated properly, it can lead to severe damage. So there are several inhibitors and inactivators that usually controls regulates the complement system. So inhibitors are the one that inhibits the pathway to continue. It will not allow the pathway to continue to cause a severe damage to the human body, whereas the inactivators are the one that completely inactivates the complement protein. They will eliminate this complement protein so that they are no longer be able to carry on the classical pathway or the alternative pathway. That is, they are not autocatalytic in nature. So among the inhibitors that are present in the human body which inhibit the complement protein are the most prominent one are the C1 esterases, which inhibit the complement protein. Next are alpha aminoglycoprotein, and you have autocatalytic prolongation proteins also that will not allow the complement protein to continue in the pathways. And we have another protein which is called as the S protein that usually inactivates the complex at a place where C6-7 complex is usually formed. And then we have inactivators which inactivates the complement protein which are no longer able to take part in the pathways. Examples are serum beta globulin of which the factor I inhibits the C3 activation, and we have beta globulin that inhibits the factor H that acts as an inactivator to inactivate the C3A spontaneous hydrolysis. And we have anaphylotoxin inactivators. Because of the overproduction of anaphylotoxin, that is a smaller unit, there can be a severe damage to the immune system. So this can be inactivated with the help of alpha globulin. And finally we have C4 binding protein that will not allow the C4 to go and bind to the bacterial surface. So these are the inactivators. So the biological effects of the complement system is that apart from bringing about the lysis of the organism, that is the cytolysis or the bacterial lysis, it also mediates other biological functions such as helps in augmenting the inflammatory response or enhancing the inflammatory response or hypersensitivity reaction because of the anaphylotoxins that are produced. It can enhance the phagocytosis by activating the phagocytes, and it can combine with the coagulation of fibrinolytic or kininogenic phagocytic system also. Now coming to the first important biological activity of this complement system is the opsonin. Opsonization is nothing but enhancement of the phagocytosis. So the complement protein has receptors on the macrophages which goes and interacts with the macrophages and enhance the phagocytosis in the macrophages. That is, they join with the antigen antibody complex and take the antigen antibody complex to the macrophages, where they give the antigen antibody complex to the macrophages and ultimately degrading the microorganisms. So this is called as opsonin. Opsonin is enhancing the phagocytosis. So apart from that, these complement system also can go and bind to the immune complexes that are formed, and these immune complexes are taken to the different parts of the body, sometimes causing a beneficial role and sometimes causing a harmful role. Harmful role in case of autoimmune diseases, but the beneficial role is they carry these complement protein along with the antigen antibody to the phagocytic cell or to the spleen to eliminate this immunocomplex or the immune complex that are formed during an immune reaction. And similar type of receptors are seen on the B cell in the EBV, that is Epstein-Barr viruses. Now coming to the inflammatory response, how this complement protein augments the inflammatory response. Inflammatory response is the response that is usually seen whenever there is a wound or damage to the body. So during that particular process, there are 4 important cardinal signs that can be seen, which are called as rubor, Calor, tumor and dolor. And there is an edema which brings more about the amount of phagocytes to that particular area. So these inflammatory responses are usually enhanced by the complement system, and thereby being more effective towards the pathogenic microorganism. And there is C2 kinins that are produced during this inflammatory response, that are vasoactive amines that helps to release various pharmacological active mediators to enhance the inflammatory response. Apart from that, we also have the smaller components, that is the subunits C3A and 5A, that are anaphylotoxins active against the anaphylactic or active during the anaphylactic responses. And next is the C567 enzyme, which is C8 convertase, and it is said to be chemotactic in nature, bring about the reactive lysis of the organism. And these complement protein also are the one that can mediate the autoimmune diseases, that is the hypersensitivity reaction, type 2 and type 3 hypersensitivity reaction. Type 2 is usually mediated by the cytotoxic cells, and type 3 are the one that are mediated by the immune complexes. So type 2 autoimmune diseases usually brings about the blood transfusion reaction. So complement proteins are also involved in this blood transfusion reaction, where there is a rejection of the blood transfusion when there is a transfer of the blood group in incompatible person. So also mediate a beneficial role to the immune system via the autoimmune disorders. And then we have the complement mediated type 3 autoimmune diseases are nephrotoxic nephritis, serum sickness, Arthur's reaction and autoimmune hemolytic anemia. So these complement system not only brings about a positive role but can also cause a harmful role in the human immune system. Now coming to the endotoxins, these endotoxins acts as an effective activators of the complement system, and they bring about the C3 fixation and the platelet adherence. Now when there is a platelet adherence with the complement proteins, they bring about the intravascular coagulation leading to thrombocytopenia. Now next, coming to the immune adherence, these complement protein goes and interacts with the antigen antibody complex and takes them to the erythrocyte. So this is called as the immune adherence. So there is cells which are in combination with the complement protein. The immune complexes which are in combination with the complement protein and erythrocytes are actively phagocytized. So there is a rapid phagocytosis that can be seen in the macrophages. And when there is a bovine serum albumin which has a specific protein which is called as K protein, called as conglutin, which goes and interacts to form a reaction with the complement protein which is coated with the complement serum protein C. So this is called as immunoconglutinin IK, which can be used in serological diagnostic procedures. Now how do you quantify that is the presence of the complements and its components in the serum? Because they are usually in inactive form, they are activated upon antigenic challenge, they can be seen as a higher dilution serum when injected with the sheep erythrocytes. So whenever there is an antigenic challenge, there is an increase in the production of these complement proteins that can be easily identified. Next way how you identify is by the hemolytic activity, whether there is an hemolysis taking place or not, because complement proteins are mostly involved in the hemolytic activity. You can identify the or you can quantify the components of complement that are present in the serum. Next is the other more sophisticated method is the radial immunodiffusion, but it is a more laborious procedure when compared to the other procedure. Next is the biosynthesis of the complement protein. How these complement proteins are synthesized and where are these complement proteins are synthesized. Mostly these complement proteins are of 20 different types. So different complement proteins are produced in different parts of the immune system. So it is in the intestinal epithelium where you can see the production of C1 complement protein. In the macrophages, the C2 and C4 are mostly synthesized, and in the spleen it is the C5 and C8, and in liver you can see the production of C3, C6, C9, and apart from these complement protein, you also have this acute phase substances. So during an acute phase reaction, these substances are mostly produced by the substances that are inducing this acute phase protein. So they are called as the proteins that are produced by these substances are C3, C5 and C6. And the deficiency of the complement system, when there is no complement system getting activated inside the body, it can lead to many type of diseases which can be sporadic in nature. And you will not see that elimination of the organism from the body, which is very effective in presence of the complement system. So the pathogen and the immune complexes cannot be eliminated efficiently in the absence of complement proteins, and there is a recurrent infection with the different types of the pathogenic microorganisms, and can lead to a genetic disorder which is called as angioneurotic edema. So mostly these edema are seen in the person who does not have this complement system activated. So this is all about the complement system that complements the antibody to bring about the lysis of the organism. Very important component which is present in the human serum.